All right, good afternoon or good morning or whenever you're watching this. This particular video takes place between two lectures. I was going to push off everything until tomorrow, Friday, and I was going to then have a weekend recording. Then I realized, why put off till the weekend what I could do right now? Not to mention we have a nice little topic to talk about right here, which is going to be how gels work. So here's the title page. We have gel separations or what's going on. Now you might have run across this depending on your exposure. Maybe you've never seen it before. Either way, we've got some of the principles that you can learn for filling in the gaps for what you know about using gels. Now, gels are different from columns. Columns can use a gel, but a gel separation is one that is fundamentally a different shape than a column separation. Columns are, like the big columns that you see in front of a Roman temple or whatever. Gels are slabs. They're just two-dimensional slabs with lanes in them. Let me show you a picture. There's a column, and right there I can tell you that that column is not an analytical column, but it's a preparative column. Basically, if you're using something for analyzing a sample and then you throw away what results at the end, that's a small thing. You want to use as small of a sample as possible because you're going to throw it away and the sample is essentially destroyed by the process. So small techniques are called analytical techniques. If you're keeping the thing that comes out the other end, whether it's a column or whether it is a gel, that's a preparative technique, and preparative techniques are going to be big. Compare this column to the columns that you used in lab. The columns you used in lab were essentially analytical columns. You were seeing what was going to happen, and yeah, you could keep different colors, but you didn't do anything with the sample after you kept the colors. Now, if you wanted to keep the vitamin B12, for example, that you isolated, and then use it for a pharmaceutical prep or something like that, that would be a preparative use of that column. But those wouldn't be very good for it because they're too small. In general, analytical small things that you throw away. Preparative is big things that you keep and you use it. They use big preparative columns. There's even ones that are I've heard they're 2 stories high. They build them into the pharmaceutical companies' factories, and they go all the way through the ceiling because they want to have the column be so large. They want to run I don't know if they can run kilograms through it, but they can probably do kilogram scale preps with a couple of runs of that column. Needless to say, that's not what we're doing, but I do have preparative columns in my lab, and they are bigger than the analytical columns. So electrophoresis is usually used analytically. Usually columns are used preparatively, although either one can be used either way. But usually you use a column and you're using it in a prep. For example, in Biochem 2, we're going to use a column to make a nice purification of the protein fragments that you design. Electrophoresis, on the other hand, is usually used analytically. The only exception to it is that you might have done a form of DNA electrophoresis where you run a DNA gel and then you cut out the lane and you dissolve the gel and you take the DNA out from it. That would be a preparative use of a gel. I'm not aware of anybody who does proteins that way. So electrophoresis, especially the way that we do it as protein chemists in my lab, is going to be analytical. And so this is what it looks like. You have a 2-dimensional array, and you have, in this case, I think it's, 12 different lanes, maybe 15. And you load this blue stuff in there. The blue dye at the top is just so you can see where you've loaded the sample. So you have 12, 15 samples all in a row, and then you turn on the juice at this point. And you can see there's a little bit of electricity going on because you see some bubbles behind it going up. You can see a little foam at the top. But what's most important, you see your sample is pulled down the gel. Now what you see for the sample is most of the lanes are invisible. But the lane on the far left has been stained already. The lanes on the right have not been stained yet, but the lane on the left is a pre-stained ladder that you can see. And in fact, I can't skip to the end again. I have a picture of what the ladder looks like on the other hand. What you will do with a gel like this is that you'll take it and then you'll put it in stain. You'll turn the protein lanes blue, and all your little proteins where they are will show up on the gel when they are. It's exactly like developing a photograph, in fact. Now that's 32 times faster than it runs, so gels can take half an hour to an hour to run. But that's sped up so that we can show you what's going on. It's like a cooking show or something like that. So the gel is a separation technique. It separates proteins. And the important thing about gel electrophoresis is it's a different type of gel than the gel in size exclusion chromatography. This kind of gel does not have individual beads. Instead, it has a tangle of strands. And so instead of having the beads, it's going to actually work the opposite of what the gel chromatography does. So the proteins will elute in a different order because what you have is you have this network of acrylamide, in the case of acrylamide link polymers. And so what you have is it's like a thicket that the proteins have to move through. And then you pull on them with electric charge, and because the thicket is so thick, they basically are pulled on by the charge, and the small molecules can wiggle their way through more easily. It's like the kids section of a ball pit or something like that. If you have the kids, they can move through the ball pit a lot faster, and they can get away from you if you're trying to catch them and things like that. It's the same kind of idea. Little things move faster because instead of having gel beads with cracks in them, you have a big gel with tangled thickets of these strands there. So it's a porous gel. The small things move farther, and so at the bottom of the gel, you'll have the smaller things. At the top of it, you'll have the bigger things. But you might be thinking about a few things about why does it work this way. Well, here's the way your book shows it working. And the important thing that you can see right there is you have some molecular weight standards in the first lane, like before. This is like the pre-stain ladder that we had on the video that I just showed. And the markers, or the ladder in this case, they're proteins that you know what they are and you know how big they are. And so you have one protein that's about 14,000 Daltons, 14 kilodaltons. And you see that you get a band right there at the bottom. Anything that lines up with that band is going to be 14 kilodaltons. And so what you can do is you can look across at this gel, and you can pretty quickly estimate how big is the protein that's being purified. Because as you go through a purification, what you're doing is you're, from left to right, you are putting the steps of the purification onto the gel. I don't think this goes exactly with the steps that we had before on the activity table that we had at the end of last lecture, but it's the same idea. You have your cells before you induce the protein, then you induce the protein, all of a sudden you see a new band in the induced cells that's right at 45,000. In fact, it's about the same exact size as the marker that we have that's at 45,000 kilodaltons that we know. And so we basically take those cells, we lyse them, and then we hopefully do a purification that gets rid of the other proteins and keeps our one protein that we're interested in. And you see it's a good purification. We have a salt precipitate, an ammonium sulfate precipitate step that already gets rid of most of the other proteins. You can see there's another one hanging around right underneath it. And so we do an anion exchange, and it gets cleaner. Cation exchange, it gets even cleaner. And then purified protein, at the very end, we have a nice thick band and nothing else right where we want it. This is a pure sample of protein, at least 95% pure. And that's usually the standard that you use for a protein prep. So what you can tell from this is that you can actually tell the size and the purity of the protein. The purification is moving in a direction from left to right in this case. So the important things about the gel chemistries, you might be thinking to yourself, if you're looking at this, well, wait a second, how are the proteins moving down the gel? And so if you look at this slide, you can see that we apply a positive charge at the bottom of the gel, and we have the proteins at the top of the gel, and so the positive charge pulls the proteins down the gel. Okay, but you can say, well, wait, Dr. McFarland, there's another issue here. Why are all the proteins the same size? If they're being pulled by the positive charge at the bottom of the gel, they must all be negative. But you just said, it depends on the pH and the PIs and things like that, but proteins have a variety of charges on their surface. How do we make all the proteins have a negative charge so that they actually move down the gel separated only by their size and not by their charge? What we do is we use the chemical in the corner up there. It's SDS, sodium dodecyl sulfate. It's a detergent. It's a good soap. It can actually be used for lysing cells. And in fact, a little bit of it is in your toothpaste, and it lyses the bacteria on your teeth when you brush your teeth. It's used for cleaning, but in small amounts. If you don't eat your toothpaste, it's safe to use in your mouth and it will destroy those bacteria. And so sodium dodecyl sulfate, you see that it's essentially a lipid because it's a long carbon chain with a sulfate on one end and then a sodium to balance the charge that will dissolve in solution to just leave the negative charge on the sulfate. So the SDS is added because it does two things at once. It actually unfolds the proteins to make them a big long chain, and it will also give them, usually you'll have like 12, 13, 14 of those attached to even a small protein. And that means all proteins will have at least a dozen negative charges on them. And so essentially what you do is you try to overwhelm the native charge of the protein so that instead it's coated with these molecules and it's coated with a negative charge. That makes all the proteins run just by size and not by charge. By the way, the gel itself is formed by cross-linked acrylamide molecules. You can change the chemistry of the cross-linking, you can do a gradient, all sorts of stuff like that. We used to pour our own gels, but now we buy them. And so the gel chemistry is not that important except that you understand it is a cross-linked gel with lots of strands. It's a thick kind of gel. So for SDS, only mass determines separation. At least that's what your reading quiz told you. Then the only problem that you have is that most proteins don't have any color to them. You can't see them. You've got this big slab where the proteins have been separated, small on the bottom, big on the top, but you can't see them at all. One possible thing would be to use a UV visualization, but that's kind of a pain. It'd be better to make the proteins visible. And so what you can do, what we commonly do is we just have a blue chemical that binds to proteins and turns blue when it sticks to the protein. It's already blue, it doesn't turn blue. Okay, and this allows you to visualize the gel. Then you see all these blue strands like what you have on this right here. And that's why most of these are blue. By the way, there is a second, more sensitive type of gel staining called silver staining. And if you see a gel that is sort of silver colored, if it's a true color picture, then that means that you may be dealing with a silver stained gel, which is much more sensitive than the Kamasi blue. But Kamasi blue works for most of our purposes. So SDS is an interesting chemical. It's important that you know what it is and what it does. And in fact, if you think about it, it's in your toothpaste, right? So it explains something that you might have noticed. If you've ever brushed your teeth and then drunk orange juice, how does it taste? Pretty bad, right? It tastes a lot more bitter, a lot less sweet. It turns out that the orange juice usually has a lot of bitter things in it. Most plants have a lot of bitter things in them, and oranges are really no exception. But it's just that usually in your mouth, you actually have a set of, I think I explained it here. Okay, yeah, you have basically a bad taste, a bitter taste, and so you will never want to have orange juice and toothpaste Lay's potato chips. These are not real. These are fake and photoshopped. They're like the worst possible flavor of Lay's. And a worst possible kind of dessert would be an orange juice and toothpaste popsicle, like right here. Okay, why does it taste so bad? It's because there are these bitter compounds in all plants. They basically make bitter compounds as a defense against insects eating them. If they're bitter, the insects don't want to eat them. But we actually have, usually we have a bunch of lipids in our mouth normally, and there's enough lipids in your mouth to hide the bitter compounds that are in orange juice. And they're actually phospholipid micelles, a type of thing that we talked about a few lectures back. But the thing about SDS is such a strong detergent that it not only disrupts cell membranes, it also disrupts these micelles that are hiding away the bitter components of the orange juice. Therefore, the bitter components of the orange juice are all getting to your tongue instead of being hidden away, and you taste more of the bitter as a result. By the way, it's a pretty good rule of thumb that bitter things have evolved to be sort of herbicides, and they've evolved to even, we taste them as bitter because we have evolved to avoid them, because they are poisonous in many cases. Now, it's a lot more complex than that, but you do have this driving force of evolution that makes orange juice taste bitter when you do it after toothpaste. Long story, but I want you to see how it all hangs together. So we can actually analyze our gels pretty closely. And in fact, if we are truly separating just by size, which for most normal proteins we are, then we can know our assigned sizes in our molecular weight standards, our ladder, our markers is the three things that's been called here. And we can take an unknown protein, and if we see this unknown protein, we can ask the question of how big is it? You can probably look at lane 2 right here and say, that unknown protein, I think it's about 40,000, maybe 39,000 molecular weight. And that could be good enough, but if you want a better estimate than just eyeballing it, what you can do is you can actually make a graph of log of the molecular weight on the y-axis versus relative migration on the x-axis. The only thing about this is notice that it is the log of the molecular weight. That's the way gels work. They separate based on the log of the molecular weight rather than the molecular weight itself. And actually, if you look at this and you think about what the distribution is, that should make sense with what you see on this gel. Because myosin is twice as big as glycogen phosphorylase, but it's not twice the gel up. It's more like a log of the gel up. Okay. This is why logs are important to biochemistry. Not many other advanced math things are. So it's mostly algebra and logs that we're dealing with. I want you to understand how you can make a standard graph out of lane 1, and then you can take the relative migration of lane 2 and use it with the line fit to figure out the log of the molecular weight of the protein in lane 2. So take a look at that if you need to pause and sort of think it through, trace it through, and look at how that works. But these are some gels from my lab. This is something that Morgan McGovern did, and she basically was using nickel affinity columns because we engineered a histidine tag onto our proteins, and we use nickel, which binds histidine really well. It binds imidazole type things really well. They associate with nickel. And so they bind to it, and then we add imidazole to elute the protein from the column, and we get a nice purified protein. And here's a wonderful gel that she ran, where she collected the different fractions, and from left to right you have fraction 1, 2, 3, and 4. The only thing we don't have on this, we don't show you where the fractions actually start and stop, but I can tell you F3, F4, F5, and F6 are in the blue box there. So you can see that we have a lot of protein coming off, and F4 and F5 look pretty good. It looks like we have a little bit of a contaminant in the bottom there, but most of the protein is a certain molecular weight, and if we know the molecular weights in the first lane, in the ladder lane, we can actually calculate the molecular weight of what F4 is. It's between that red rung on the top and the rung right below it. So here is another fragment. So the full length one comes off in a gel like that. The fragment comes off in a gel like this. And if you look very closely, you can see that the fragment is a little bit lower on the gel than the full length, which is exactly what it should be. A fragment is smaller than the full length protein. You can actually solve this problem. If you look at the different colored, and because the gel itself was pre-stained, they pre-stained the 75 kilodalton and the 25 kilodalton proteins pink. So they got to be pink, everyone else was blue. But you can see from there where the 75 is. You're below 75 for both of them. And you can actually try to estimate what is the difference between the full length on the right and the fragment 4 on the left. Also notice about this, all gels run a little differently. That's why you need an internal standard. You need this ladder because it looks like the gel on the left was run a little longer than the gel on the right. But because you have that ladder of standard sizes, you aren't going from just the absolute movement, you're going from the movement relative to the standards, okay? And you can see from that, you could even calculate it. I'm not asking you to calculate it, but I'm asking you just to estimate how much smaller is fragment 4, do you think? And this is a biochemistry joke right here. They have the Mickey Mouse gene, which is a triple repeat of the 8 amino acid motif, methionine, isoleucine, cysteine, lysine, glutamate, tyrosine, methionine, serine, glutamate. That is a joke. It might not be a very funny one, but it's a joke that actually will help you use your biochemistry knowledge. Use what you know about those to turn that amino acid sequence into the single letters versions of it, and you'll see what it says to you, okay? And the thing below it is an entire joke. It's an entire fabrication down there. That there's no such thing as a protein with mouse ears. Okay. The other thing is I want to tell you sometimes, the first level of knowledge, you can say something like what your book said, which is that gels separate proteins by molecular weight alone. But I'm here to tell you, when you go out into the real world, you'll find that the textbook doesn't lie. It just tells you what's true 95% of the time. And then there's 5% of the time where life throws you a curveball. And here's a case where sometimes you actually have the gels behaving in a complex manner. Here's this report right here, and what they're saying is they're actually saying that there are some acidic peptides that don't follow the rules. They didn't read the textbook, and what they're doing is they're moving in a way that makes sense if you think about it. But they noticed something funny. The gels weren't following the textbook, and so they wanted to figure out why. This is what they noticed. They're running this DEF protein that migrates is supposed to migrate to 87 kilodaltons. And you see right there they've done a little graph where they show you where the molecular weight standards were. It's supposed to be between the 95 and the 72. But instead you can see right there it is above the 95. It's around 100 kilodaltons. They have a difference in molecular weight that is 13.2 up. And so their question is why? So they chased this down. They did the experiment. They made all these different fragments. The full-length DEF protein is 750 residues long. So what they did is they made all these fragments that they show here from like residue 190 to residue 753. Then they cut off at residue 378 to 753. So they cut off the N-terminus on the first couple, and then they cut off the C-terminus on the last ones, the ones that are labeled with a 2. And you notice something funny about when they ran it on the gel. When they ran it on the gel, the original protein has a delta molecular weight. It is about 13 kilodaltons bigger than it should be. So it's at 12.8 on that graph E. But if you notice the N-terminal truncations, the ones where they cut off the N-terminus, they run just fine. They don't have any deviation from the expected molecular weight. However, all the fragments that had the N-terminus in it, they all had that deviation as well. So this allowed them to zero in on the problem. They knew that there was something in the first 200 residues that's making the protein stay farther up on the gel than it should. So you can see that these fragments are the ones that are weird and are wrong and are running on the gel different from what you expect. So it must be some N-terminal residues in there messing things up. Therefore, what they did is they looked at the sequence. And then the first thing that you want to think about is if you have something that is farther up on the gel than it should be, then basically it's not being pulled. You suspect there's a charged residue thing going on. Now, because they're supposed to be coded by these SDS-Page things, you can have a complex relationship. So I'm not going to predict whether it's going to be negative or positive charges, but I will predict that there could be a charged residue either way. So let's look at the N-terminus and see, are there a lot of basic residues or a lot of acidic residues? And if you light up all the acidic residues pink and look, there's all those E's and D's are only acidic residues, and they make up like half the protein. And so they looked at this and they said, I bet that it's these acidic residues that are messing up how the gel runs. And they actually went back and they actually did a graph of what percentage of residues were D or E and how big the deviation was from expected values of molecular weight. And it turns out if 50% of your residues are D or E, you'll have a 100 Dalton movement of your molecular weight that they see here. And so they made a whole graph here where you can actually fit that to a line, and they worked out an equation that they had in the abstract where you can see if you have a high percentage of acidic amino acids, you can expect your gel, your protein, to run too far up on the gel. So look at all the other ones. They separated out by strongly basic residues, hydrophobic, polar, and other. And none of those show a straight line like strongly acidic does. So that's kind of cool. Now, if you think about it a little bit, The acidic residues mean that you have more negative charges, which should mean at first blush that you have it more pulled toward the positive end. But the other thing is those negative charges will also mean it will bind less SDS, because it will repel the SDS. So you actually will have less SDS binding, and that is probably why you have it running farther up on the gel than you expect, because you have less negative charge of the SDS loading onto the gel. We're not even exactly sure if that's the reason why. All we know is that there's this observation, and it doesn't fit the simple pattern of the charge on the protein affecting how far down it runs on the gel directly. There must be some kind of indirect effect. So that's the evidence for how they chase down. This only is important if you have really acidic proteins. And it will only work for that, it only caused a problem for that particular protein. So usually the textbook is right. Most proteins are not 50% acidic amino acids. But if you're working with an acidic peptide or something like that, you should be aware that sometimes these physical effects can overwhelm what the textbook explanation for the average protein is. There's one more kind of gel and then we're done. This isn't really, this is a separate kind of gel, but it can be used with the SDS-Page kind of gels that we do. By the way, PAGE stands for polyacrylamide gel electrophoresis. This kind of gel is IEF for isoelectric focusing. And the one thing about this is it's exactly like something we've already talked about. We already talked about how isoelectric focusing works for amino acids. It separates them by a charge along a pH gradient until they stop at their pi. So what happens with this is it's the exact same thing we already talked about, just applied to proteins rather than amino acids. The important thing about this is you need the proteins to be at their native charge. So you better not add SDS because that's going to mess up their charges. It's going to slap all these negative charges on the proteins, then it'll mess up everything. But if the proteins are at their natural charge and you load them at one end of a strip with a pH gradient through the strip, and then you pull on them with a positive charge, they will move according to that positive charge until they reach the pH where the pH equals the pi, and just like for amino acids, when the pH equals the pi, charge on the sample is neutral. A neutral sample is not moved by an electric charge, and so they stop moving. So this really isn't even a new kind of gel. It's a kind of gel, a technique that we've already talked about, just applied to proteins in a gel format. But what's cool about this is that you can, just like with the amino acids, you can read out what the protein's pi is. The PIs that are very basic are going to be on the left in this representation. The PIs that are very acidic will be on the right. And now, what somebody had a great idea was, if we have these two techniques for gel, one separates by molecular weight, the other separates by charge, I wonder if we could do both. When in doubt, do both. There is something called two-dimensional gel electrophoresis, where you take a protein sample, you put it onto an isoelectric gel where it separates in the side to side, it separates side to side by the isoelectric point, the pi. Then you take that strip and you load that onto the top of a 2D gel. And then you pull down on it, you add SDS, by the way, at this point, and then you pull down on it is separated by molecular weight. So separated by pi in this direction is separated by molecular weight in this direction, in the y-axis direction. And so you literally have something that really spreads out all your proteins. You can actually run an entire lysate on this, and you'll have a messy separation, but it will be a true separation. You can see how many proteins are at which pi. And so, for example, you look at this, and like this cell has a lot of proteins, has a lot of small proteins that tend to be at the basic pi. So it has a lot of small basic proteins, not as many big acidic proteins. And so you can see what this looks like, and you can even, in some cases, you can actually take the protein out of the gel, and then you can actually run it through a mass spec. More on that in the next lecture. But the thing about this right here is that you usually can't identify what the proteins exactly are without a little effort. But what you can do is, let's say that this right here is the two-dimensional gel of a normal cell. Then you take the same cell, but when it's been transformed into a cancer cell, a tumor. And you take that and you lyse it and you run it on two-dimensional gel electrophoresis. what you'll see is you'll see a lot of the proteins that are the same, but you'll see some proteins that are different. And then you can go after those proteins. You can try to purify them. You can try to sequence them. You can try to say, this protein is there in cancer, but it's not there when the cell is normal. I wonder if that protein might be part of causing cancer. Or it might be part of an effect from cancer, and you can learn something about cancer from that. So even though these are big and messy, they are actually very useful for determining what's going on in a complex disease like cancer. There is actually a little simulator. It's in this this paper. You actually have access to the paper through the library, but you don't need to read it. Mostly, I just want to show you that they do a little simulation of where would all the proteins in a cell separate if you ran them on a 2D gel. And they do this nice little simulation. You just see there's a bunch of different proteins, but notice what they choose. In the drop-down menus, you choose your range of pH for the x-axis. You choose your range of acrylamide concentration for the y-axis. The more acrylamide, the thicker the gel, and you'll have different molecular weights separating out based on that. And that just shows you, again, these are these two axes. Two-dimensional gel electrophoresis has PIs on the x-axis. It has molecular weights on the y-axis. And you can get a lot of proteins onto one gel. And you can start to see some things from that. If you want to see what this looks like, you're free to look it up, but I'm not going to make you. You've got enough to worry about. Focus on Chen et al., of course. That brings us to our homework assignment. So yes, this is our little gel interlude, and hopefully I'll be able to fit everything into the lecture tomorrow, and then we'll be ready for the test. So I will see you either Friday if you happen to be watching this really quickly, or I'll see you Monday to review the test if you're watching this over the weekend. All right, take care, everyone.